Okay.
All right.
Yep. Go back to your slides. Sorry
All
for the
right.
interruption.
No problem. It's my fault for being late. So is to use a relatively recent technology of vision transformers. And in our case, to apply those to cropped regions of the full-size image that contain individual cells. And we'll see more about that. So the idea is take the multiplex image, segment into individual cells, and then crop out individual cell regions, and then apply the vision transformer to learn a model to predict the cell type from the intensities across that cropped image in all of the channels. And we trained this model, and we'll talk more about it in a second, using a very large collection of already available multiplexed images from various tissue types. And in order to be able to use that diverse database, we consolidated the labels to a set of common 17 cell types, rather than having some of the data sets might have even finer resolution labels, but those aren't present in other images. So we consolidated it down to 17 major cell types, including most of the immune cells. Okay, now here's the next thing. What we decided to do, because we wanted to be able to apply this across a range of antibody or marker panels, we separated images into different cohorts and trained models for different combinations of markers, so that we would have, a very high resolution immune model, and a lower level and a lower level, and then we had a model for nerve cells and so on, right? So we had 5 different models that were trained with this large data set, and these we made into an ensemble model so that any given image would be brought in and then could be applied to one or more of those panels. Now, for each of those, what happens is that we take a patch around an individual cell. We apply a soft masking to remove the parts of other cells largely from that region, so that we're focusing in on just this cell in the center of that region. And then the idea behind the way the vision transformer works is that we randomly mask different regions of that cropped cell and train the encoder and decoder to be able to reconstitute the full cell image. And thereby it is learning a representation of the pattern in space of a particular cell type. So that is what gets mapped then to the label for that particular cell type. The original paper on vision transformer is shown there. All right. So combination of training separate models, each of which can learn to recognize different cell types, and this base approach to building each of those models using vision transformer. Now, a significant problem that sometimes requires retraining of models is that different antibody panels that people choose don't have the same markers as other panels that other people have trained models on. And so the traditional way of dealing with that is to just add in a blank image for the missing marker, kind of hope for the best, okay? And that may work if that marker is basically can be substituted for in the overall prediction task by some other markers. But what we thought we would try to do is to augment the ability that the model has to try to learn that on its own by, when markers are missing, to impute the missing marker by using another vision transformer, such that the input to the model would be as close to complete as it could be, okay? And I'll just show you that approach works. By the way, I should have mentioned, the way that imputer works is that you're masking out in the training of that imputer different channels so that it can learn to fill in the missing channel, okay? So that's as opposed to the other approach where you're masking out panels. Okay, so the interesting thing was that this worked. So if we compare for 3 different of these models, right, the sort of minimal one, the middle immune model, and the full, if we compare the performance of the model on test images that it hasn't seen before, when we give it all the markers, or we eliminate one of the markers, blank it, or we eliminate that marker and then impute it using the approach I just described, okay? And when that happens, what you can see is that for the base, by losing 1 or 2 of the markers, we have significant drop in the accuracy of the classification of the cell types. But if we do the imputation, we preserve most of that accuracy. And that happens across all of the panels. In fact, for the immune full panel, we can eliminate as many as 4 markers and still get almost the same results as we would if we had them. And fundamentally, that's saying there's a lot of redundancy or some level of redundancy within these panels, but that even so, it's better to do the imputation than just to hope that the model will figure it out on its own. Okay, so using this approach, we have then this ensemble of 5 models and the imputation facility. And so we applied that particular model, that ensemble model, to 2 large collections of images, one of which was a fresh collection of Human Protein Atlas tissue microarray images. Many of you may be familiar with the Human Protein Atlas. And then also to a collection of codex images from the HubMap project. So here are some examples of tissue microarrays to which we have applied this RIPCA and assigning the different cell types. And, you know, of course, you see, depending on the tissue, you see different compositions of the cell types. But more important than just showing you a picture and saying, you know, look, it's pretty, we check to see how a couple of aspects of that. One of which was to try to determine how much it mattered, how much the accuracy of the cell segmentation mattered to this process, okay? Because if you're doing the vectorized approach where you take the cell and you just convert it into a vector of marker intensities, if you're making mistakes in the segmentation, then that vector is going to be off from what it should be. Whereas, ideally, for the spatial approach, it can sort of deal with that. It can say, well, okay, you know, I'm not quite sure if this is the right boundary, but I can still tell what the cell type is, all right? And so what we did is we ran segmentation masks in different ways, randomly, and then applied the RIPCA to each of those different segmentations, and you can see they're quite different, and then asked how far off were we from estimating the cell types in those differently segmented images. And what you can see is for basically all of the cell types except the epithelial cells, the error is minor, it's really minor. So it's able to compensate for errors in the segmentation by using this spatial approach. Okay, so of course, we then needed to show that not only did we get pretty pictures with cell types assigned, but that those are accurate to whatever extent we trust human annotation to be accurate. And so what we did is we chose example images at random and then had a human annotator go in and say, is this the correct one or not? And what you can see is that, again, in the confusion matrix there, with the exception of smooth muscle, where there are more errors and we're still working on trying to fix that, there's very high accuracy or very high agreement between the human annotator and the predicted cell types. By the way, on the lower left there are comparison of the results to PIXI, which is a more unsupervised approach to trying to assign cell types, or just doing k-means clustering of the intensities and just choosing clusters based on that. RIPCA does better than both of those. So again, here are the results of running this over 46 different human tissues. And what you can obviously see is that different tissues differ in their cell type composition. And we also applied the same approach to a large collection of hub map images. In this case, multiple images within a cell type, and you can see consistency and variation from image to image from the same cell type. Now, the last point that I want to make is, of course, once you have assigned cell types, then you can proceed to assign larger elements, larger regions. And so in this case, what we did is straightforward clustering of a region by the cell type composition of the region, but then asked, again, expert human annotators to say, what's that orange region that I'm showing up here as a cluster, right? And what you can see is that we identify very specific types of structures that are known to be present in those tissues. So last thing, there's a Napari plugin that will allow you to load your images and run this. By the way, I should have said the model will, I think I skipped over that. The ensemble model will match up whatever markers you have in your panel, and then decide, am I close enough of a match to this that I can then do inference to fill in the rest, or am I just missing the markers that I need for that panel, in which case it will just skip over that. And then it forms a consensus decision from the models as to what it provides. And what the plugin will do is also to allow you to annotate the results if you want to try to get an estimate of the accuracy or whatever. All right, so that's the story of RIPCA. I should very strongly mention the sources of funding. This was a collaboration between my group at Carnegie Mellon and the group. Okay, I can see her, Emma Lundberg. That's it. This is what happens when you get old. An Emma Lundberg's group at Stanford and prior over in Sweden. And you can see there was a lot of work that was put into this, and that was supported by these agencies. So I'll stop there, and thank you very much for your attention.
Thank you so much. We will maybe take one question before we move on to the other talk.
Thank you, Professor Murphy, for the really nice talk. I just had one quick question. So, were you able to find a bound of sorts up to how many missing markers was the model able to work well with? Yeah. So
I showed you what we did, right? In that we picked a random marker, left it out, and for the smaller panels, once you remove two, you're not having a lot left. And so that's as far as we went. Larger panel, we were able to do 4. It would be easy to just extend that, but we just didn't go that far. All right.
Thank you so much, Dr. Murphy.
Thank you.
Thank you again.